All right, so we're going to move into the next learning targets of Unit 3, the second half, which is learning targets 11 through 12. So we ended learning target 10 talking about the light-dependent reaction and the light-independent reaction, or the Calvin cycle. So this learning target, or these two learning targets, are going to dive into those with a little bit more detail. Now, this is a longer presentation with a lot more information in it. I'm going to streamline it in these videos because you can pause it and go back and watch as many times as you need to. And it typically takes us 2 days in class to get through these two learning targets. We'll just try to do the light-dependent reaction on one day and the Calvin cycle on the other day. So if you want to tackle it that way with your video watching as well, that is 100% fine. So the chloroplast is where all of this is going to take place, and this is the organelle and plant cells and a few bacteria where photosynthesis can take place. The chloroplasts have their own DNA, ribosomes, and consist of a double membrane, and this gives way to the endosymbiosis theory that we talked about back in section 1 of Unit 3. So the thylakoid are flattened discs inside the chloroplast where chlorophyll and other pigments are located. The light-dependent reaction occurs on the thylakoid membranes. A couple different structures of the thylakoid. You have the lumen, which is a space inside the thylakoid, and a grana is a stack of thylakoids inside the chloroplast. More surface area allows for more work to be done. So if we go back one slide real quick, a stack of these is a grana. So the stroma is a fluid inside the chloroplast and outside the thylakoid. So the Calvin cycle occurs in the stroma. Light-dependent reaction. So the light-dependent reaction is a reaction of photosynthesis that requires sunlight to make ATP and NADPH. And remember, NADPH is the electron carrier that has picked up excited electrons. Okay, those electrons come from a magnesium atom in chlorophyll that, when it's hit with sunlight, it excites those two electrons that are picked up by NADP plus, turning it to NADPH and carrying it over to the Calvin cycle. So you'll see half of photosynthesis reaction, the light reaction here. Water comes in, oxygen's a byproduct. Okay, oxygen's a waste product of splitting of water. So steps of the light-dependent reaction. Light hits the pigments of photosystem 2, causing magnesium to lose its outer two electrons, and these electrons will enter the electron transport chain, and it's often referred to as the ETC. The sunlight at PS2 is photosystem 2, also causes a water molecule to split, releasing the electrons into the photosystem that replenish the electrons that are lost. H plus ions are also released into the lumen. The oxygen is given off as a waste product. Now, this is something important for us to understand. Now, you understand that plants need water to live. All living things need water to live. But water plays an underlying role in all of this, okay? Magnesium becomes excited when sunlight hits it and loses two electrons. But the problem is magnesium doesn't have an everlasting supply of electrons to lose. So in order for this system to happen more than once, those electrons have to be replaced. The splitting of water is what replaces the electrons that are lost when magnesium is hit with sunlight. So while water, yes, is needed, we need to understand what it is that water does for the process of photosynthesis. And it replaces the electrons that are lost when magnesium is hit with sunlight. And it also creates a concentration gradient of H plus ions that is going to give us the ability to, or give the plants ability to do work. Remember, we talked about concentration gradients creating the ability to do work. So keep that in mind as we move through this and we get to that H plus concentration in the lumen. All right, so the electrons travel through the ETC, providing power for H plus ions to travel across the concentration gradient into the lumen. The concentration gradient is building in the lumen, which is creating the ability to do work. Remember, anytime we're pumping ions across the concentration gradient or against it, it is a form of active transport. So energy is needed. So, the old saying is you have to spend money to make money. Well, the same thing holds true here. You have to spend energy to make energy. The electrons come to photosystem 1 and are struck by light again. The electrons are re-energized at this point. An electron carrier called NADP+ grabs the two electrons and creates NADPH. NADPH will now travel to the Calvin cycle. Okay, so sunlight hits magnesium at photosystem 2, exciting electrons. Those two electrons leave, go through photosystem 2, powering proton pumps to pump H+ ions against the concentration gradient. Okay, they travel to photosystem 1. By the time they get to photosystem 1, those electrons have lost their energy. They're hit with sunlight again to excite them. After they're hit at photosystem 1, NADP+ grabs them, creating NADPH, and that is what's going to be sent to the Calvin cycle, or the light-independent reaction. So now let's get to the concentration gradient of the H+ ions in the lumen. So they will diffuse across the thylakoid membrane through specialized proteins called ATP synthase. There's that ending again, ASE. Remember, we talked about proteins a lot of times have that suffix for names. So ATP synthase. As protons, H+ ions, move through the protein, it spins. The spinning action creates energy to add a phosphate group onto ADP, creating ATP. And that ATP is sent to the Calvin cycle for the creation of sugars. So the spinning of ATP synthase, that protein, will provide the energy needed to attach a phosphate group to ADP. Okay, remember, in order to create a chemical bond, energy must be put into it. So actively creating a concentration gradient of H+ ions or protons, okay, that is going to provide the ability to do work to provide energy to make this compound. Remember, nature does not want concentration gradients. So as the cell creates this concentration gradient in the lumen, okay, nature's going to want to even it out. So once the concentration gradient is great enough, those protons are going to move through ATP synthase, spinning it like a top, creating the energy as it moves towards equilibrium. So summary, the reactants of the light-dependent reaction are water, NADP+, and ADP. The products are oxygen, which is given off, NADPH, which is going to go to the Calvin cycle, and ATP, which is going to go to the Calvin cycle. So in class, this would be where we would stop today, and we would pick up with the Calvin cycle tomorrow. So however you want to handle this, for those of you watching this video, that's up to you. The Calvin cycle, or the light-independent reaction, is a reaction in photosynthesis that requires ATP and NADPH to convert carbon dioxide molecules into high-energy glucose molecules. All right, so the steps of the Calvin cycle, a little bit more nitty-gritty, a little bit more terminology-based than the light-dependent reaction. So 3 molecules of carbon dioxide combine with 3 molecules of a 5-carbon sugar to make 3 6-carbon molecules. This is called carbon fixation. Okay, carbon fixation is a way to say that carbon and carbon dioxide is changed from inorganic carbon into organic sugar molecules. So the carbon and carbon dioxide is inorganic and it cannot be used by living organisms. It has to be fixed. Rubisco is the enzyme that combines the carbon to the 5-carbon sugar. Rubisco is the most abundant protein on Earth. Those 3 6-carbon sugars quickly split into 6 3-carbon sugars called PGAs, and those are what's going to drive this process. Each PGA molecule is energized by 1 NADPH and 1 ATP. So if there's 6 PGAs, then total from the light-dependent reaction we're going to need is 6 NADPHs and 6 ATPs. NADPH loses the electrons it gained from the light-dependent reaction and becomes NADP+. That NADP+ will return to the light-dependent reaction. ATP loses the phosphate group that it gained in the light-dependent reaction from ATP synthase and becomes ADP and returns to the light-dependent reaction to be re-energized by ATP synthase. The 6 PGA molecules are now named G3Ps. The energy caused them to change their shape, thus they changed their names. So the PGA molecules are unenergized. Once they become energized by the products of the light-dependent reaction, their name will change to G3P. So of the 6 energized G3P molecules, one, that shouldn't say on, it should say one is set aside for later. There are 5 G3Ps left. The remaining 5 G3P molecules need to return to the original 3 5-carbon molecules that we started the process with. 3 ATP molecules are used to rearrange these G3P molecules into the original 3 5-carbon molecules. Once this happens, the cycle will begin again. Once this cycle occurs twice, we now have 2 G3P molecules that have been set aside. Each time this process happens, 1 G3P is going to be set aside. Those 2 G3P molecules will combine to become a glucose molecule. So summary, 3 carbon dioxide molecules will give us 1 G3P or 1 Calvin cycle. 2 G3P molecules or 2 Calvin cycles was needed to create 1 glucose molecule. The reactants of the Calvin cycle are carbon dioxide, NADPH from the light-dependent reaction, and ATP from the light-dependent reaction. And the products are glucose, which is what is used for energy in living things, NADP+, which is an empty electron carrier, which is heading back to the light-dependent reaction to pick up more electrons, and ADPs, which is a adenine diphosphate, which has lost its 3rd phosphate group, which is now going back to the light-dependent reaction to become re-energized. So the rate at which this process occurs, both the light-dependent reaction and the light-independent reaction, there's different things that can control the rate. One of those is temperature. The higher the temperature, the higher the rate of reaction can be. However, if the temperature becomes too high, the reaction will stop. And the opposite is true for being too cold. Water. If there is a low amount of water, the reaction will not occur because the electrons cannot be replenished. Carbon dioxide concentration. At high concentrations of CO2, the rate of reaction increases but levels off at a certain concentration. And last but not least is light intensity. At high light intensities, the rate of reaction increases but levels off at a certain concentration. So there's a little bit longer of a video, but that's where we're going to end for learning targets 11 through 12. Feel free to go back, pause, rewatch, email me questions if you have it, and be sure to complete the video form that goes with this presentation. If you need anything, let me know. Thank you.